Next is about amphibian development. So we are slowly moving on to the developmental biology portion. So fertilization can occur in the animal hemisphere of an amphibian embryo. The point of sperm entry mark the ventral side of the embryo, while the side 180-degree opposite the point of sperm entry, cortical cytoplasm rotates 30 degree with respect to the inner cytoplasm. So the inner gray cytoplasm in the marginal region called the gray crescent mark the dorsal side and beginning of gastrulation through producing dorsal lip of blastopore. So animal cortical cytoplasm is a pigmented site. Animal pole is there. Sperm entry point will be there. Vegetal cortical cytoplasm which is unpigmented. Then vegetal pole will be also there. So the cortical cytoplasm rotates relative to the inner cytoplasm. So cortical cytoplasm rotates relative to the inner cytoplasm. And the gray crescent is created by rotation. Next is about amphibian gastrulation, vegetal rotation and the invagination of the bottle cells. On the future dorsal side below the equator in the region of gray crescent, the cells invaginate to form a blastopore and cells change their shape called bottle cells that line the archendron. So gastrulation in frog begins not in the most vegetal cells like sea urchin but in the marginal zone, the region surrounding the equator of the blastula, where the animal and vegetal hemisphere meets. The vegetal rotation takes place. The perspective pharyngeal endodermal cells adjacent to the blastocele and immediately above the involuting mesoderm, involuting marginal zone IMZ. So involution occurs at the blastopore lip, where the dorsal lip of the blastopore becomes composed of cells that become the prechorodal plate, the precursor of head mesoderm, and chorda mesoderm cells that form the notochord. With the formation of ventral lip, the blastopore has formed a ring around the large endodermal cells that remain exposed on the vegetal surface. This remaining patch of endoderm is called yolk plug. The animal cells undergo epiboly and coverage at the blastopore. Convergent extension of the dorsal mesoderm. As it continues, it narrowing the mesodermal layer and lengthen the involuting marginal zone. The convergent extension of mesoderm appears to be autonomous. Towards the end of the gastrulation, the centrally located notochord separates from the stomach mesoderm on either side of it, and the notochord elongates separately as its cells continue to intercalate. Those cells entering through the dorsal lip of the blastopore give rise to the ventral dorsal mesoderm notochord and somites. The remainder of the body mesoderm, which form the heart, kidney, bones that enter through the ventral and lateral blastopore. The endoderm is derived from the superficial cells of the involuting marginal zone that form the lining of the archendron roof. The remnant of the blastopore, where the endoderm meets the ectoendoderm, now become the anus. Then progressive determination of the amphibian development. Unfertilized amphibian egg has polarity along the animal-vegetal axis. The animal hemisphere blastomere will become the cells of the ectoderm, skin and nerves. The vegetal hemisphere cells will form the cells of the gut and associated organs, endoderm, and the mesoderm cells which form the internal cytoplasm around the equator. So once the dorsal portion of the embryo is established, the movement of the involuting mesoderm establishes the anterior-posterior axis. The first endomesoderm to migrates over the dorsal blastopore lip will induce the ectoderm above it to produce anterior structures like forebrain. So mesoderm that involute later through the dorsal lip of blastopore allow the ectoderm to form more posterior structures such as hindbrain and spinal cord. Primary embryonic induction by mesoderm. Then enhance Spearman inductive interactions in regulative development. When the fertilized egg of the newt was constricted by a ligature, the nucleus was restricted to the one half of the embryo. At the 16th celled stage, a single nucleus entered in the individual in undivided half. After 14 days, each side has developed into a normal embryo. After first cleavage, when the cleavage plane be aberrant, the grey crescent material passes into only one of the two blastomeres and develop normally, and the other blastomere produce a mass of unorganized tissue lacking dorsal structures. When the egg is divided along the plane of first cleavage into two blastomeres, each of which gets half of the grey crescent, so both cells develop into normal embryo. So normal development only proceeds when the cellular constriction occurs along the correct plane, because the embryo is already asymmetrical as seen with the grey crescent. Then determination of ectoderm during newt gastrulation. Neural ectoderm from one newt embryo is transplanted into a region in another embryo that normally become epidermis. When the tissue is transferred between early gastrulate, the neural tissue develops into epidermis, so the cells of the early gastrula were uncommitted and exhibit conditional development. So when the same embryonic tissue is transplanted into late gastrula tissue, the neurula cells cause neural plate, so cells are determined, and the transplanted cells exhibited autonomous development. Then Hans Spearman and the Hildmann-Gold primary embryonic induction. Of all the tissues in the early gastrula, only one has its free, autonomously determined, and a dorsal lip of blastopore, the tissue derived from the grey crescent cytoplasm. When this tissue is transplanted into presumptive belly skin region of another gastrula, it can initiate gastrulation and embryogenesis in the surrounding tissue. In an experiment, they visualized the process by which, using two differently pigmented newt embryo, dorsal lip tissue from an early T. tenuitus gastrula is transplanted into T. cristatus gastrula in the region that normally becomes ventral epidermis. The donor tissue invaginates and forms a second archindron and then a second embryonic axis. Spearman referred to the dorsal lip cells and their derivatives, notochord head ectomesoderm, as the organizer because they induced the host ventral tissue to change their fates to form a neural tube and somates. They organized host and donor tissues into a secondary embryo with a clear anterior-posterior axis. How does the organizer form? The next topic, determination of ectoderm during newt gastrulation. So neural ectoderm from one newt embryo is transplanted into a region in another embryo that normally becomes epidermis. When the tissue are transferred between early gastrulate, the neural tissue develops into epidermis, so the cells of the early gastrula were uncommitted and exhibit conditional development. When the same embryonic tissue is transplanted into late gastrula tissue, the neural cells causes neural plate. So cells are determined and the transplanted cells exhibited autonomous development. Then Hans Spearman and Hildmann-Gold primary embryonic induction. Of all tissues in the early gastrula, only one has its fate autonomously determined, that is dorsal lip of blastopore, the tissue derived from the grey crescent cytoplasm. When this tissue is transplanted into presumptive belly skin region of another gastrula, it can initiate gastrulation and embryogenesis in the surrounding tissue. In an experiment, they visualized the process by which two different pigmented newt embryo, dorsal lip tissue from an early T. tenuitus gastrula is transplanted into T. cristatus gastrula in the region that normally becomes ventral epidermis. The donor tissue invaginates and forms a second archindron and then a second embryonic axis. Spearman referred to the dorsal lip cells and their derivatives, notochord head endomesoderm and the organizer, because they induced the host's ventral tissues to change their fates to form a neural tube and somates. They organized host and donor tissues into a secondary embryo with clear anterior-posterior axis. So, how does the organizer form? The cells that form organizer positioned opposite the point of sperm entry, get two signals that tells the cells that they are in dorsal position, and second signal says that they are mesoderm. Then the dorsal signal beta-catenin. The dorsal lip of blastopore obtained its properties from the induction provided by the vegetal cells. Newcoop removed the equatorial cells, presumptive mesoderm, from a blastula showed that neither the animal cap, that means presumptive ectoderm, nor the vegetal cap, presumptive endoderm, produced any mesodermal tissue. But when the two cap cells are recombined, the animal cap cells are induced to form mesodermal structures such as notochord, muscles, kidney, and blood cells. The polarity of induction depends on whether the endodermal fragment was taken from the dorsal or ventral side. Lateral vegetal cells choose close to the point of sperm entry induced ventral mesenchyme blood and intermediate mesoderm, while the dorsal most vegetal cells specified dorsal mesoderm components. Somites notochord that have properties of organizer called Newcoop center. The major candidate for the factor that forms the Newcoop center is these vegetal cells. This beta-catenin experimental depletion of this molecule results in the lack of dorsal structures, and injection of exogenous beta-catenin into the ventral side of embryo produces a secondary axis. Beta-catenin is initially synthesized throughout the embryo from maternal mRNA. It begins to accumulate in the dorsal region of the egg during the cytoplasmic movements of fertilization and continues to accumulate at the dorsal side throughout the cleavage. During the first cycle, when microtubules forms parallel tracks in the vegetal portion of the egg, GBP migrates towards the growing end of the microtubule opposite side of the sperm entry, future dorsal side, by binding to kinesin along with dissolved by the process of cortical rotation. At the dorsal side, GBP and DSH inactivate GSK3, allowing beta-catenin to accumulate and cause organizer-specific gene activation, but on ventral side, the beta-catenin is degraded. So another paracrine factor, WNT11 mRNA, is located in the ventral cortex during oogenesis and translocated in the future dorsal side by cortical rotation of the cytoplasm. The WNT11 signal stabilizes the GBP and DSH and organizes them to protect beta-catenin. Beta-catenin combines with the ubiquitous transcription factor known as TCF3, converting the TCF3 repressor into an activator of transcription. In the organizer region, dorsal side, the beta-catenin and TCF3 completely appears to bind with the promoter of twin and siamosis gene. If these genes are ectopically expressed in the ventral cells, the secondary axis emerges on the ventral side of the embryo. In the ventral side, the TCF3 protein inhibits siamosis and twin transcription when it binds to the promoter region of the genes within the absence of beta-catenin. Siamosis and twin proteins bind to the enhancer of several genes involved in organizer function, along with SMAD2/4 transcription factor activated by vegetal TGF-beta B family members, nodal related proteins, PG1, actin, etc. Together, this TFS activates the organizer specific genes associating goosecoid XLIM1 and paracrine factors and agonists like Nogin, Cordin, FRZB, Cerberus. The presence of VEGTTF in the endoderm prevents the organizer genes from being expressed outside the organizer area. Goosecoid essential for specifying the dorsal mesoderm. Goosecoid expression occurs when there is synergism between siamois twin and TGF-B signals secreted by vegetal cells, and the vegetal TGF-B like a signal. Along with beta-catenin pathway, the TGF-B signal from vegetal cells critical in activating the genes that characterize the organizer cells. Two maternal RNAs tethered to the vegetal cortex that are crucial for the ability of the vegetal cells to induce the cells above them to be mesodermal. VG1 and VGT are the two TFs that instructs the endoderm to synthesize and secrete TGF-B cells family members activating diarrhea and several nodal proteins. Each of them can activate the XBRA gene encoding a TF that instructs the cells to become chroma mesoderm that become the autocode. Activin can induce different types of mesoderm at different concentrations. Those regions with little nodal related proteins across the endoderm induce the cells above them to become ventral mesoderm. Regions above vegetal cells with some nodal related proteins become lateral mesoderm, and regions above vegetal cells containing large amount of these proteins become the organizer. Functions of the organizer. The ability to self-differentiate dorsal mesoderm, precaudal plate, chorda mesoderm, etc. The ability to dorsalize the surrounding mesoderm into paraxial somite forming mesoderm when it would otherwise form ventral mesoderm. The ability to dorsalize the ectoderm and induce the formation of neural tube. The ability to initiate the movement of gastrulation. Cells of the organizer contribute to the four cell types: pharyngeal endoderm, forebrain and midbrain; precaudal plate, head mesoderm, forebrain and midbrain; dorsal mesoderm, hindbrain and trunk; and dorsal lip of blastophore, tip to tail. And proteins expressed in the organizer. Nuclear proteins. Twin siamois, the XLIM1, OTX2, glucosid HNF3B. Secreted proteins: chordin, Dickoff, Adinf, then Noggin, Follistatin, Sonic Hedgehog, Cerberus, nodal related proteins. Introduction of neurula ectoderm and dorsal mesoderm, BMP inhibitors. It is the epidermis that is induced to form not neural tissue. The ectoderm is induced to become epidermal tissue by binding of bone morphogenetic proteins. While the nervous system forms from that region of the ectoderm, it is protected from epidermal induction by BMP inhibiting molecules secreted from organizer. Default meet of ectoderm. Three of the major BMP inhibitors secreted by the organizer are Noggin, chordin, and follistatin genes product. Then Noggin. Noggin is a secreted protein that is able to accomplish two major functions of the organizer. It induces dorsal ectoderm to form neural tissue. It dorsalizes mesoderm cells that would otherwise contribute to the ventral mesoderm. Noggin binds to BMP4 and BMP2 and inhibits their binding to receptors at the ectodermal cells. Injection of Noggin mRNA into one cell UV radiated embryos complete rescued dorsal development and allow the formation of complete embryo. Then chordin. Of all organizer genes observed, chordin is one of the most acutely activated by beta-catenin. Like Noggin, chordin binds directly to BMP4 and BMP2 and prevents complexing with their receptors in the dorsal lip of blastophore and autocode. Then follistatin. Follistatin mRNA is also transcribed in the dorsal lip of blastophore and autocode. Follistatin is an inhibitor of both activin and BMP, cause ectoderm to become neural tissue. Then epidermal inducers, the BMPs. In Xenopus, the epidermal inducers are bone morphogenetic proteins BMP4 and its close relatives BMP4, BMP7 and ADMP, anti-dorsalizing morphogenetic protein. If the mRNA for BMP4 is injected into Xenopus egg, all the mesoderm in the embryo become ventrolateral mesoderm. BMP4 induces ectodermal cells to become epidermal, but organized secreted proteins like Noggin, chordin, etc. prevents BMPs from binding and inducing the ectoderm and mesoderm cells near the organizer. So, BMP4 is expressed initially throughout the ectodermal and mesodermal regions of the late blastula. During gastrulation, BMP4 transcripts become restricted to the ventrolateral margin zone because of fuscoid protein that repress the transcription of BMP4 and WNT8. In the ectoderm, BMP repress the genes such as neurogenin involved in forming neural tissue while activating other genes involved in epidermal specification. In the mesoderm, the graded levels of BMP4 activate different sets of mesodermal genes, thereby specifying the dorsal, intermediate, and lateral mesodermal tissue. So BMP4 may explicit expression of different genes in the concentration dependent manner. So the region of codon Nogin BMP4 is totally prevented from binding and these tissues become notochord. Slightly further away from the organizer, the MYF5 gene is activated that form dorsolateral muscles. As more and more BMP4 molecules are allowed to bind to the cell, the XEV-ENT2 endolateral and ventral genes become expressed. Then the regional specificity of neurula induction. The organizer tissue not only induces the neural tissue, it also specifies the region of the neural tube, that which part of the ectoderm will differentiate into forebrain, mid-hindbrain, and spinal cord. The most anterior part of the archindron roof containing head mesoderm induced balances and portions of the oral apparatus. The next most anterior section induced the formation of various head structures including nose, eyes, balances, and optic vessels, vesicles, and third section including notochord induced the hindbrain structures, and most posterior section induced the formation of dorsal trunk and tail mesoderm. The pharyngeal endoderm that underlies the head secretes Dickoff and FRZP block WNT proteins, nodal related proteins, and BMPs. The precaudal plate secrete the WNT blockers Dickoff and FRAB as well as chordin, nogin, and follistatin. The notochord contains BMP blockers chordin, nogin, and follistatin but does not secrete WNT blockers. So IGF from the head endomesoderm probably act at the junction of the notochord and precaudal mesoderm. Then two major gradients in the amphibian gastrulation, i.e. BMP gradient that specifies the dorsal ventral axis and a WNT gradient that specifies the anterior posterior axis. Retinoic acid is also seen to have a gradient highest at the posterior end of the neural plate. So RI signaling is important in patterning the hindbrain and appears to interact with FGF signals to activate posterior Hox genes.